begin?
So hello everyone, my name is Eloise Müller. I am currently a postdoctorate associate at the University of Albany in the US. But today I'm going to present to you some work from my PhD that I did at the University of Paris-Saclay in France. Okay. So during my PhD, I was interested in parasitoid wasp and their associated polyidnaviruses. So parasitoid wasp is a wasp that needs a host to spend its development in. On this first picture, you can see an example of a wasp that injects its eggs in a caterpillar. And about 2 weeks later, you can see the wasp larvae that go out of the caterpillar. Of course, the caterpillar usually does not survive such a parasitism. What is very interesting here is that at the same time as the eggs, some of those wasps also inject polyidnaviruses, which are domesticated viruses. Once in the caterpillar, those polyidnaviruses can express virulence genes and inhibit the immune response of the caterpillar in such a way that the caterpillar cannot defend itself against the invading wasp larva. So the polyidnaviruses, which I'm going to call just PDV, is actually a paraphyletic group. Here is a very simplified tree of some parasitoid wasp, which are called the ichneumonids, and who are composed of 2 families of wasps, the braconids and the ichneumonids. And you can see here represented in blue that there was a major event of endogenization of a virus that took place about 100 million years ago in a braconid wasp. That was then followed by the domestication of some of those viral genes. And nowadays, all the descending species have those domesticated viral genes that allow them to make PDV. Independently, you can see that there was other independent event of endogenization of another virus this time, but in the same way, it was also followed by domestication. And here again, the descending species are able to make PDV. When the PDV is found in the ichneumoniid genome, we call it intact ichnovirus. Whereas when it is found in the braconid genome, we call it a bracovirus. During my PhD, I mostly focused on the bracovirus of a single wasp species, so braconid wasp species. So let's take a closer look on the structure of those bracoviruses. As I say, bracoviruses are domesticated viruses, which means that they are found in the genome of the parasitoid wasp, represented here in yellow. And they are composed of 2 elements. The first element are the nudibranch genes. So those are the genes that were domesticated 100 million years ago, and that encode for viral particles. The second element of bracoviruses are the proviral segments. Here I show only 2 of them, but you can have, like, for example, 30 segments in the wasp genome. You can see that each of those proviral segments is delimited by this motif called DRG for direct repeat junctions that allow each proviral segment to excise from the wasp genome and to circularize into DNA circles. In the wasp females, those DNA circles are amplified in multiple copies and packed in the viral particles. It is this whole thing which is injected in the caterpillar at the same time as the eggs. So here we have some wasp DNA which is directly injected in the caterpillar. So we can wonder what happens to those DNA circles once in the caterpillar. And mostly, could they integrate in the genome of the host, which will correspond to horizontal transfer from the wasp to the caterpillar? The short answer is yes. Some studies already shown that at least some DNA circles of at least some parasitoid wasp were able to integrate in the genome of the caterpillar, represented here in green. However, little is known about the mechanisms of these integrations and about the frequency of such integrations. So the first goal of my study was to better characterize the integrations of those bracovirus in the caterpillar host. To do this, I focus on the wasp species Coccidia typhae, whose only natural host is Sedamnia non-icoides, also known as the Mediterranean corn borer, who is a major pest. And what we did is that we parasitized 18 of those caterpillars, and we dissected them 7 days later. We could observe wasp larvae in 12 of those caterpillars, which means that parasitism succeeded. However, we could not observe any wasp larvae in the 6 other caterpillars, which means that parasitism failed. We were mostly interested in the 12 caterpillars in which parasitism succeeded, so we dissected for tissues or structures from those caterpillars: the head, the ganglionic chain, the hemolymph, and the fat body, on which we extracted DNA. And then we sent those 4 samples to Illumina sequencing. In addition of those 4 samples, we also have a 5th sample that corresponds to the whole body of a caterpillar in which parasitism failed. So when we received the raw Illumina reads, we had to look for chimeric reads. So what is a chimeric read? If I show you again the step of the DNA circles integration in the genome of the caterpillar, you can see that if we sequence such an integration, most reads will map on the caterpillar genome in green, some reads will map on the wasp genome in yellow, and some reads will overlap the junction, so those ones will map on both genomes. Such a read is called a chimeric read, and counting the number of chimeric reads, we are able to estimate a number of integration events for each of the 5 samples for each of the DNA circles of Cotesia typhae. So this is the main result. Here you can see the normalized number of integration events for each of the samples. So the first thing to see is that we do have integrations in all the tissues we investigated. This is actually the first evidence of integrations elsewhere than in the hemolymph, which shows that most tissues seems to be accessible by those bacuviruses, even though the hemolymph seems to be the tissues with the most integrations. Another very interesting result is that we found integrations even in the sample called whole body. If you well remember, this is the sample in which Possum fails, which means that this caterpillar could have survived to adult age if we would not have killed it for sequencing, and it may have reproduced. So after this result, we have been wondering whether surviving caterpillar could transmit such integrations to the offspring, which will correspond to a successful hostile transfer from the parasitoid wasp to the Lepidoptera host. Sadly, I don't have time to go more into details about the mechanisms of this study. So I'm directly going to go to goal number 2, in which I investigated whether the bacuvirus integrated in the Lepidoptera could be transmitted throughout the generations. So in order to be transmitted, 2 things have to happen. 1, the integration had to take place in a cell that will be transmitted, so in a germinal cell. And 2, this integration had to persist until the adult stage. So here we firstly check the second point. So you can see in red the number of integration at the caterpillar stage, so just after parasitism. And in blue and green, you can see that at least some of those integrations were able to persist until the adult stage of surviving caterpillars. More precisely, we could estimate that there was 2 integration events per haploid genome in average in adult number 1, so twice more per diploid cells. And in adult 6, we even found 12. So this sounded like quite a lot to us. From those numbers, we could expect from 12 integration events per gametes in average. If this number are true, we should be able to detect transmission to the next generation very easily. So this is what we try to do. For this, we crossed some surviving moths to see if they could transmit any of those bacuvirus integration to the offspring. For this, we sequenced 500 offspring. However, we could not detect a single chimeric read in those offspring, which means that we were not able to detect any transmission. So how come with such a high frequency of integration? We could think of 2 possibilities. The first one might be that gametes are actually not as accessible by bacuviruses than the other tissues. And the second explanation may be that they're as accessible, but the ones with too many integrations are not functional. So we didn't sequence those ones through the offspring. They were not transmitted to the offspring. So Ines Matruki, who is a student in our laboratory, tried to go a little further on this question. So for this, she firstly sequenced some gonads of male that survived parasitism. And she was able to show here that in her 3 replicates, there were integration in the gonads. So this shows that gonads are accessible by the bacuviruses, even though you can see that there are way less integrations in gonads than the hemolymph. However, gonads are composed of both somatic and germinal cells. And here we have no way of knowing in what cells those integrations took place. So after this, she used a method to sequence only the sperm cells of the males. And here, she could not find any chimeric reads. So taking all those results together, we can conclude that we could not detect any transmission because the gametes are not, or at least very little, accessible by bacuviruses. So those results do not prove that transmission is impossible. What it shows is that if it's possible, it takes place at a frequency which is way too low to be detectable by our pipeline. Yet, at the scale of evolution, even a very rare event can take place many times. So in this last goal, I investigated whether we could find any trace of hostile transfer in the evolution of Lepidoptera. To answer to this question, we stopped to work on just a single wasp species and a single Lepidoptera species. Instead, we work with all the genomes of Lepidoptera available on NCBI. So at the stem of the study, there were 775 genomes, which represents 35 families of Lepidoptera. So here you can see the phylogenetic tree of those 35 families. And here you can see more precisely how many species were included for each of the family. So you can see it's very biased. For some family, we have only one species, whereas for the Hesperidae family, we had more than 300 species. And then what we did is that we did some similarity searches, thanks to BLAST-N, between all those genomes and sequences of Pulidnaviruses. So here we chose to use both Brachyvarus and Ichnovirus sequences to have a better idea of the impact of the Pulidnavirus on the evolution of the Lepidoptera. So this is the exact same figure as previously, but now the colors shows you what we found or didn't find in each of those genomes. So in purple, when a species is in purple, it means that we could not detect any trace of Pulidnavirus in this genome. When it is in orange, it means that we found at least one trace of Ichnovirus in the genome. In green, at least one trace of Brachyvarus. And in brown, it means that we found traces of both Ichnovirus and Brachyvarus in the same genome. So here you can see that most families seems to be implicated in those horizontal transfer. And overall, we found that there was 16% of the genomes with traces of Pulidnaviruses. So here, of course, we refer of Pulidnaviruses in the all cells of the organism, so something that has been transmitted throughout the generations. So to conclude, we firstly showed that Brachyvarus which are integrated in the genome of the caterpillar are very little transmitted to the next generation, at least in the Brachyonid Cotesia typhae that we studied very precisely. However, at the scale of evolution, we could find a lot of traces of Pulidnaviruses in the genome of Lepidoptera, which suggests that Pulidnaviruses is a major vector of horizontal transfer from parotid wasp to their Lepidopteran host. So to finish, I would like to thank all the collaborators of these studies. And of course, thank you for your attention.
All right, so we've got plenty of time for questions. If you have a question, please either use the raise your hand function, or if you would prefer to do it in writing, there's the question and answer feature within Zoom. All right, so I will ask you a question. So if you had additional time to keep working on the same system, what would you like to look at next?
Yeah, so there are like so many things. So there is first the part which is really just on horizontal transfer, which was what I was the most interested in at the beginning. So the next step will be to see if there are also traces in other species than Lepidoptera, because this is the main host, but that's not the only one. There are many other insects. Then, I mean, we show in terms of frequency that there are a lot of transmission, but we're not able yet to show what is the real impact on the evolution. Like, do those Pulidnaviruses can have a function? Can they be domesticated by the Lepidoptera? So this will be very interesting to look at.
All right, thank you. And Francisco, you have a question as well.
Sí. Muchas gracias, Eloisa, es muy interesante el tema. Y mi pregunta es muy general. La transferencia horizontal es muy importante para pues, la evolución en sí, ¿no? Pero quiero saber qué piensas acerca de la transferencia horizontal y la simbiosis. Recientemente estoy viendo trabajos acerca de que se han descubierto bacterias en las vacuolas de levaduras. Entonces, no sé tú qué piensas acerca de esto, si es una simbiosis. Bueno, son microorganismos, pero que se integran a otros genomas finalmente, ¿no? Entonces, ¿qué te parece esto respecto a tu tema en general. Y bueno, en general, pues todo. Si alguien tiene algo que comentar acerca de esto que me interesa de momento, se los agradezco. Gracias.
Yeah, so I haven't activated the captions, so I don't really know what the question is. Did anyone?
The question
But,
was
yeah.
asking you if you want to speak about the importance of horizontal gene transfer with symbiosis in general, relating to some of their work with bacteria and yeast.
Yeah, so this would be, we already know that there are quite some horizontal transfer between bacteria, these bacteria and animals, but we know very little about horizontal transfer between two animals. So this was what I was really interested in. So there are very few examples of horizontal transfer between animals. And indeed, symbiosis is one of the big hypotheses saying that species in symbiosis could have more transfer. And this is a very good example of such a case. But there's not a lot of other study about other symbiosis.
All right, at this point, we're going to move on to our next speaker. Francisco put in the chat that he actually prefers to play the video that he recorded in English. So we're going to do that. He is here in the session, though. So if you have questions, feel free to type them into the question and answer, and he can answer them while you do so.
Please in English. Hello, partner of the Third Young Congress on Evolutionary Biology. My name is Francisco Garcia Franco of Mexico City. The topic I now present is entitled A New Model of Hybrid Incompatibility by Metabolic Control Reveals the Origin of Overdominance and Inbred Depression, an antecedent of Darwin's corollary. Based in findings we found in 3 studies, 2 experimental and 1 bioinformatic. Important implications arose on fundamental paradigms of evolutionary theory, such as the relative amount between male and female fertility genes of each is architecture in the genome as a product of heterozygous and homozygous recessivity suffered by the genes which are integrated respectively. The Haldane role, the Darwin's corollary of asymmetric offspring on reciprocal process, and the origin of the overdominance, subdominance, inbreed depression, and heterosis. On the other hand, through a deep review of evolutionary theories, we are able to build new mechanisms and direct models that explain the paradigms of evolutionary theory in a more adequate way. Introductions. In 1858, Charles Darwin and Alfred Earle officially presented the theory of evolution by natural selection, which describes the biological process of how new species arise from the slow accumulation of heredity factors that, under the daily structural force of the Bible, individuals with greater variability are fitter and consequently are favored by natural selection. Subsequently, beginning in the 1920s, with the resource of evolutionary theory as a neo-Darwinism, a large number of scientific researchers, such as Ronald Fisher,
It looks like your video just stopped. All right. E, we can't hear anything. Did you share the computer sound?
Of the been inflated with the new knowledge that is emerging. Of the models and the theories integrated into synthetic theory of evolutions, the Mendel's theory of genetic segregation and dominance provide the genetic basis that got the evolutionary theory the greatest robustness and structure. This allowed a large number of models explaining different speciation processes, such as the model of speciation, both allopathic and ecological barriers, the changing equilibrium theory, even the neutralist theory of evolution, considered as an alternative model to the process of natural selection, as well as the dominant Bateson-Dosomsky-Mueller model of hybrid incompatibility. They are based on Mendelian dominance. Parallel to the development of evolutionary models based on Mendelian dominance, models and theories basadas en metabolic control have been developed fueron de amongst the gene product of the two alleles of each gene are additive and contribute to the total productivity of the gene. But in the same time, each gene is a link in the gene change or pathways that determine the functionality of cellular process, where each gene contributes individually to maintain the overall flow of the pathways the which is apart, producing a robust global flow, even with the appearance of the recessive mutation of loss of function in an allele of one or more genes, with the consequent decrease of up 50% or more in the productivity of the affected gene or genes, which ultimately determines a level of dominance that could keep the phenotype close to the wild type. The correct studies and theory that support the theory or dominance of not metabolic way, the mutation in the genes of the glycolytic pathway producing recessive mutant phenotype manifested as pathological disorders due to hemolytic anemia, HI, and homozygous loss of function mutation in any genes at the melanogenesis pathway produces the mutant phenotype of albinism. For other times, no gene works alone. They are grouped into modulus of interconnected genes that determine each of the common cellular process. Many there are redundant. Biallelic expression is predominant diploid genomas. Each of the two alleles of each gene produce a differential expression that is additive to the total productivity of the gene. Results. We made 3 studies of segmental integration more adequadamente expressed architecture of reproductive isolation due to hybrid sterility. 1. Heterozygous introgress subsegments larger than 30 to 40 produce male sterility. Homozygous introgress subsegments of size between 2 to 50 only produce male sterility, plus 2 or rather than 50 in size, they produce sterility in both sexes. 3. More female fertility genes than male fertility genes that can mutate into sterility. They are randomly interspersed between the genes of common cellular process along all chromosomes. It takes more female fertility genes than male fertility genes to produce the respective sterility. For new evolutionary models based in the theory of dominance by metabolic control. The model of the genetic architecture of the phenotype genetic exteriority during interspecific integration events of sister species. 352 total number of genes in the D1A, D5D region that include interrecessive segments D1A, D5D, produce sterility in both sexes. D3A, A1H produce male sterility. The threshold number of fertility genes that must be introduced into heterozygosity or homozygosity to produce sterility of the respective sex. For example, D1A, D5D produce female sterility, have 20 female sterility genes, color pink, 8 sterility genes of both sexes, yellow color, a total 28 genes. D3E E1H male sterility segment have 8 genes for male sterility, pink color, 7 sterility genes of both sexes, yellow color, have 15 total genes. For other way, the origin of the asymmetrical descendants hybrid in the parental crosses of pure species between male Dicophary with female Dicophary, panel A, and between male Debusari with female Debusari, panel B, as well as in their respective reciprocal crosses, all offspring are viable and fertile. While interspecific parental crosses between male Dicophary and female Debusari, panel C, it does not produce offspring. And in their respective interspecific parental reciprocal crosses between male of Debusari with female Dicophary, panel D, asymmetry in the offspring is manifested with semi-viable males with sterility and survivors and viable and fertile females as a direct property of the complementarity between the different combination of testicular genes coming from both parents. Conclusions. 1. There are almost twice as many female sterility genes as there are male sterility genes. 2. Integration of a minimal threefold amount of sterility gene is required, which is higher for female sterility. 3. The greater robustness of female sterility is a consequence of the greater number of female sterility genes. 4. Sterility is caused by the decrease in additive gene productivity in a manner equivalent to recessive mutation that with integration in homozygosity is more severe than in heterozygosity. 5. Our finding the segmental and genetic architecture of hybrid sterility, the greater number of female sterility genes, the greater the robustness of female fertility, the decrease in gene productivity recessivity are more adequate to understand with a direct and mechanics models based in theory of dominance by metabolic control. Thus, the offspring of parental cross and hybrid descendant segregate equivalent genotype arrangements with variable dominance that revolve the origin of overdominance, subdominance, and recessivity of inbreeding depression, as well as the asymmetry of the viability and fertility of the offspring of the reciprocal crosses, Darwin's corollary. Each of them associated with their respective phenotypes, close to or far from the ideal wild phenotype or the ideal mutant phenotype. The team is el Jacome Galindo Pérez, Blanca Estela Chávez Sandoval, Francisco García Franco, and Lilian Milena Barandica Cañón. Thanks. This bioinformatics study was carried out from in Mexico during and after the confinement due to the COVID-19 pandemic since 2020. We also thank lawyer Eva Beatriz García Franco, Professor Estela Sandoval Gómez, and colleagues Blanca Estela Esel Jacome Galindo Pérez for the financial support for my personal maintenance and for the stock resource, which allowed me to dedicate myself full-time during of these years to the research work, analysis of results, and the complete writing this manuscript.
All right, so we now actually have to switch over to our next speaker. I will be setting my speaking language to Spanish, so you should be getting captions whatever you've set your caption language to. If you have questions for our previous speaker, Francisco, you can put them in the question and answer, and he can answer them while the next talk is playing. So let me share my screen. And share my sound. Hold on just a moment. My Zoom controls moved when I did that, and I need to make sure I got the caption set up correctly. Actually, I need to stop my sharing for a moment just to set my captions.
Vale.
Buenos días, yo soy Blanca Estela Chávez Sandoval y les voy a presentar el trabajo Obtención de introgresiones cromosomales en homocigosis revelan la arquitectura genética de las barreras de aislamiento reproductivo. Aquí tenemos el método de squash de cromosomas politénicos, que es descrito desde 1881. Y en el panel A tenemos los cromosomas que son homólogos. En el panel B tenemos cromosomas no homólogos y se ve que están desacoplados. Estos son de la descendencia de Debussati con D. Keffer después de la F15. Y en el panel C tenemos un solo segmento introgresado que está en la sinapsis que se obtuvo después de 15 cruzamientos. Aquí están los cruzamientos parentales, en el panel 2, parentales de Debussati con De Keffer, donde las hembras son fértiles y los machos estériles es lo que siempre se ha visto. En el panel 3 tenemos machos híbridos contra hembras híbridas y no producen descendencia. En el panel 4 tenemos machos parentales de Debussati con hembras híbridas y no producen descendencia. Sin embargo, cuando tenemos machos parentales de Dequefere con hembras híbridas, las hembras son fértiles y los machos son estériles. En esta diapositiva tenemos en el panel A los cruzamientos que se realizan tradicionalmente para obtener segmentos introgresantes en heterocigosis, y en el panel B el trabajo que nosotros realizamos para obtenerlos hasta homocigosis. Estos son los resultados donde observamos que los parentales Bussati siempre son fértiles, en heterocigosis pues mayoritariamente también, pero en homocigosis ya tenemos muchos casos en los que son estériles. Estos son los 6 segmentos que analizamos en este trabajo y los porcentajes de cada uno. Los segmentos en rojo tenemos los segmentos que producen esterilidad, que son más del 40% para el cromosoma 4 o deben ser del más del 40% para dar esterilidad. Semiesterilidad, segmentos del 30 al 40% y fertilidad, segmentos pues menores al 30%. Sin embargo, en homocigosis, pues son estos otros porcentajes. Como conclusión, todo este trabajo nos ha permitido obtener la arquitectura del aislamiento reproductivo en estas dos especies de Dequefere y de Bussati, y tenemos unos umbrales mínimos para la esterilidad en machos de aproximadamente del 20 al 30%, para fertilidad del 30 al 40% semiesterilidad y mayores al 40% esterilidad o letalidad. El umbral mínimo de segmentos introgresantes en homocigosis es del 2.24% para machos y del 15% para ambos sexos. Y pues como se había visto anteriormente, hay mayor número de genes de esterilidad en hembras. Este gran número de genes de esterilidad explica la robustez de la fertilidad femenina, y esto también tiene implicaciones para la regla de Jalani. Aquí tenemos al equipo, al maestro en ciencias de CEL, al doctor Francisco García Franco, a la doctora Lilian Barandica Cañón y una servidora, Blanca Estela Chávez Sandoval. Agradezco a todos su atención y quedamos a sus órdenes para cualquier comentario. Gracias.
So this speaker's video was very short, so we have plenty of time for questions. Who would like to go first? All right, I guess I will ask. So why do you think it is that this particular species shows such a different pattern of the number of introgressed genes between males versus females from what you've seen in other species?
No, en otras especies no lo hemos visto, solo en
Pero sí,
Dequefere y
está reportado
Debussati,
en todas las especies, sí,
pero pues está reportado en todas las especies del género. No sé
And
si
E, I thought I saw your hand go up as well.
Yes, so I'm wondering, what do you have planned next for this research?
Pero,
Ah, pues
¿ven?
ok.
Bueno, también el doctor Francisco va a responder la pregunta.
Ok, tenemos que seguir en el paso siguiente, que es evaluar todas nuestras introgresiones en homocigosis en todos los cromosomas. Este patrón, tradicionalmente, históricamente, desde el año 86, ya ha sido reportado para todos los cromosomas de este par de especies hermanas, de Busati y de Keferet. Y se ha visto este mismo patrón para la gran mayoría de experimentos que se han hecho con el género de pares de especies hermanas de Drosophila. Incluso el mismo patrón se repite en plantas, en otros insectos, son patrones evolutivos muy generales. Entonces, lo que sigue ahora es como desde 1986 hasta ahora hemos logrado de poner por primera vez en homocigosis estas introgresiones de manera importante nos dieron el hallazgo, los hallazgos, que ahora vemos que son contradictorios a lo previsto anteriormente. Ahora tenemos que comprobar lo que está sucediendo en todos los cromosomas, aunque ya la teoría nos está dando la luz de que sí es el mismo patrón en todas las, por lo menos en todo el género ya experimentado de Drosophila.
All right, anyone else? We still have another minute or two left for questions. Well, if you come up with another one that you can ask in the question and answer, the speakers are here and able to answer them there. But meanwhile, we'll move on to our last speaker of this session. So Paiwei, did you want to share your screen?
Sure. Should I start now or should we wait until 15?
You can share your screen now. We're gonna wait until we hit the 15 to actually have you start talking though.
Sounds good.
So, you got 2 minutes and 15 seconds.
Yeah, to be nervous for 2 minutes.
If you would like, we could stare very intently at our computers for you. All right, your wait is over. You should feel free to start talking.
Cool. Hello, everyone. My name is Pei Wei Chen. I just got my PhD 2 weeks ago from Caltech, so yay. I'm excited to be here today to tell you some of my more recent unpublished graduate work, and I hope you'll like it. I think many of us here know that the genome is the battleground where different types of selfish genes compete for inheritance. On one hand, we have overproliferators like transposons, which are these mobile genetic elements that can move around the genome to increase their copy numbers. With a higher copy number of themselves, transposons will have higher chance to be inherited to the next generation. But their activities or their jumping behaviors are detrimental for the host genome because they cause DNA damage and genome instability. On the other hand, we have transmission disorders like sperm killers, which are selfish genes that distort equal transmissions between homologs to bias their own inheritance. For example, in the male meiosis, we start from one diploid cell that divides twice to generate 4 haploid cells, which then differentiate into mature sperm. According to Mendel's law, each haploid gamete carries either one of the 2 homologs. So for sex chromosomes, for example, each sperm either carries the X or the Y chromosome, and both the X and Y have 50% chance to be inherited to the next generation. But if selfish genes on the S chromosome try to cheat, they can find ways to selectively kill the Y-bearing sperm, thereby increasing the chance of S chromosome to be inherited to more than 50%, sometimes almost 100%. This is a phenomenon that we call meiotic drive, which violates Mendel's law. Even though selfish genes are so diverse, many of them cause fertility defects, so they must be silenced to protect host reproduction. In addition, many selfish genes are also often lineage-specific, meaning they're different in different species. But if selfish genes are so diverse and they look differently in different species, how can we control them? In animals, many selfish genes are controlled by a conserved genome defense mechanism called the piRNA pathway. At the core of the piRNA pathway, we have a small non-coding RNA called piRNA, shown in dark blue here, which can belong to the pIRI protein, shown in light blue. The piRNA can bring the pIRI protein to find complementary target RNAs and silence expression. In this case, the piRNA is the specificity factor that identifies the target, while the pIRI protein is the effector that silences the target. Such a binary core architecture of the piRNA pathway implies that the system is very programmable, because by changing just the piRNA sequences, the entire pathway can be used to control different targets. For example, in the fruit fly, Drosophila melanogaster, which is the organism I work on during my PhD, there are 2 pIRI proteins called alpha-NAGO3, which are endonucleases that can cleave target RNAs to silence the expression. And their targets include different transposons and a selfish meiotic driver called stellate. Silencing this diverse array of selfish genes is absolutely essential to fertility. Now, the binary core architecture I told you about of the piRNA pathway is actually not unique to the piRNA pathway. In fact, this architecture is shared by many other genome defense mechanisms. For example, in eukaryotes, there's another small RNA pathway called the siRNA pathway, where the siRNA identifies a target, while Agar protein silences the target. In bacteria, the guide RNA identifies the target, while Cas9 protein silences the target. In many tetrapods, we have different Krafft finger proteins, or Krafft DFPs, each of which can recognize different transposon sequences. But the silencing of those transposons is mediated by a shared cofactor called Cap1. Perhaps because of this conserved binary core architecture of many different genome defense mechanisms we see across the tree of life, where a new specificity factor can be easily innovated to recognize and control a new target. When we think about how to control lineage-specific selfish genes, people have always focused on the innovations of these specificity factors I'm showing you in dark blue. In other words, our current understanding of genome defense against lineage-specific selfish genes has been largely limited to innovations of specificity factors. But I wonder whether genome defense might require other types of innovations beyond specificity factors. Outside these factors that recognize the emerging selfish genes, we do not really know much about how lineage-specific selfish genes are controlled. To approach this question, I focus on a recently evolved selfish gene called stellate, which is only found in Drosophila melanogaster. But despite their recent emergence, silencing stellate is absolutely essential for fertility. The stellate RNA is recognized by recently evolved suppressor stellate piRNA, which is also only found in Drosophila melanogaster, but it protects fertility. In this case, the innovation of a new piRNA, a new specificity factor, allowed the conserved piRNA pathway protein machinery to recognize and control stellate. But ever since the discovery of this piRNA over 20 years ago, people have kind of assumed that controlling stellate is done solely by the innovation of this new piRNA. We do not know any other innovations they require to control stellate. But because stellate has such a strong impact on the reproductive success, and the suppressive stellate pirates are actually exceptionally abundant, we hypothesized that there might be other types of innovations that are required to control stellate. So I decided to look for other unknown repressors of stellate, which might contain other types of innovation. So we did a genetic screen to look for unknown repressors of stellate. And the screen was done by an extremely talented undergraduate in UNIS who did in situ to visualize the expression of stellate. As you can see, stellate is completely silenced in wild-type testes, but highly expressed when it's non-silenced. Excitingly, UNIS screen identified one hit, a novel genome defense factor that is required for stellate silencing. We named this gene Trailblazer because we like to think that it's a pioneer that controls recently evolved selfish genes over evolution. Consistent with the fact that stellate silencing is absolutely essential for male fertility, we found that males that don't have Trailblazer almost never produce any progeny, so they're completely sterile, while females don't really seem to care whether they have Trailblazer or not. Importantly, such male infertility we see upon Trailblazer knockdown can be rescued if we also knock down stellate at the same time, suggesting that stellate repression is the cause of male infertility we see upon Trailblazer knockdown. I want to take a step further and ask, how does Trailblazer facilitate stellate silencing? How does it work? What is the mechanism? To answer this question, I profiled a transcriptome of males that don't have Trailblazer and found about 30 genes that differentially express across the entire genome, which include the strongly derepressed stellate genes. Interestingly, 2 genes that are downregulated are actually ALP and AGO3, the 2 endonucleases I told you earlier that can cleave stellate RNAs to silence the expression. But if ALP and AGO3 proteins themselves have no target specificity, why is stellate silencing particularly sensitive to the loss of ALP and AGO3? What I haven't told you is that stellate is a multi-copy gene that can have over 200 copies in the genome, so the transcripts are extremely abundant, which likely require a large number of ALP and AGO3 proteins to efficiently silence. Consistent with this idea, the male infertility we see upon Trailblazer knockdown can also be rescued if we express additional ALP or AGO3 proteins, suggesting that Trailblazer facilitates stellate silencing by promoting the expression of ALP and AGO3. But how does it do this? How does it promote the expression of ALP and AGO3 genes? When I image the Trailblazer protein, I found that it's a nuclear protein that binds chromatin and actually forms foci on DNA. So I wonder whether it directly binds the promoter of these 2 genes. And this is indeed the case, because when I did ChIP-seq for Trailblazer, I found about 3,000 binding sites across the genome. Excitingly, the 2 strongest binding sites of Trailblazer are actually promoters of ALP and AGO3, suggesting that Trailblazer is a transcription factor for these 2 genes. It's satisfying to figure out the mechanism of how it works, but I wonder how did it evolve? And this is why I'm presenting at this evolution meeting. I mentioned earlier that stellate is a recently evolved selfish gene only found in Drosophila melanogaster. So initially, I was really hoping that Trailblazer would also be a young gene that was born much recently. But instead, Trailblazer turned out to be a much older gene that can be found in many fly species. So I wonder whether it contains or reflects evolutionary adaptation in order to control these lineage-specific selfish genes. One way to check this is to ask whether an ortholog from a closely related species can functionally substitute Trailblazer in Drosophila melanogaster. So we use CRISPR-Cas9 to replace the Trailblazer gene by a close ortholog from a sibling species, Drosophila simulans, that diverged less than 3 million years ago. And this was done by another talented undergraduate in Catherine, who was truly a CRISPR wizard. After we replaced the Trailblazer gene by the stimulans allele, we found that it actually failed to silence stellate, and this led to complete male infertility, suggesting that a close ortholog that diverged less than 3 million years ago is unable to functionally substitute Trailblazer in Drosophila melanogaster. Importantly, these fertility defects can also be rescued if we knock down stellate or express additional ALP or AGO3 proteins, suggesting that these fertility defects we see result from the inability of stimulans Trailblazer protein to activate sufficient ALP and AGO3 proteins to silence stellate. In other words, within the likely recent adaptive changes within the Trailblazer protein, they are required to upregulate ALP and AGO3 to silence stellate and protect fertility. But I wonder, where are these adaptive changes within the Trailblazer protein? In other words, where does evolution act on Trailblazer? The Trailblazer protein has a structured N-terminal region and an unstructured middle region, both of which can activate transcription. It also has a zinc finger domain and a C-terminus, which presumably binds DNA. In order to identify the critical evolutionary changes within the Trailblazer protein, we work with a population geneticist at UC Irvine, Grace Lee, to perform the McDonald requirement tests to look for signals of positive selection within Trailblazer. Essentially, we look at 100 amino acid sliding windows along the entire gene. Excitingly, we identified a single region within the DNA binding domain that shows signals of positive selection. Specifically, we see an excess of non-synonymous substitutions that are fixed between Drosophila melanogaster and Drosophila similans, suggesting a history of adaptive evolution within the DNA binding domain. In order to empirically interrogate the functional impacts of these evolutionary changes, we use CRISPR-Cas9 to replace each of the 3 domains of Trailblazer protein by orthologous sequences from Drosophila similans. Excitingly, replacing just the DNA binding domain was both necessary and sufficient to disrupt stellate silencing, suggesting that critical evolutionary changes within the Trailblazer protein occur within the DNA binding domain. Now, putting everything together, we think ancestrally, when stellate first emerged, through the innovation of a new pIRA that can recognize stellate, the conserved pIRA pathway protein machinery was able to recognize and suppress stellate. But the selfish stellate gene family undergo a radical copy number expansion, producing extremely abundant transcripts that ultimately surpass the silencing capacity of the pIRA pathway. We think this is where the Trailblazer protein came in. With its recent adaptive changes within the DNA binding domain, it was able to upregulate alpha-negro 3 proteins, thereby silencing abundant stellate RNAs and protect male fertility. In this sense, we think Trailblazer is a higher-level modulator, which adjusts the amount of general defense machinery to match the abundance of selfish genes. And to answer the question I posed earlier, within genome defense does require innovations beyond specificity factors. In the case of stellate silencing, in addition to generating a new pIRA, innovation within the Trailblazer protein is absolutely essential to upregulate alpha-negro 3 to silence stellate. We think tuning the general defense machinery to match selfish genes abundance might be a recurrently employed strategy in genome defense. Today, I told you our discovery of the Trailblazer protein, which tunes the pIRA pathway to tame expanded stellate genes. But in Drosophila similans, there's actually a different selfish meiotic driver gene family that underwent a recent copy number expansion, and they are recognized by recently evolved siRNAs. In many tetrapods, we see different families of retrotransposon undergoing lineage-specific copy number expansions, and they are controlled by recently duplicated Krabs zinc finger proteins, Krabs ZFPs. While people have always focused on identifying individual siRNAs and individual Krabs ZFPs to recognize the emerging selfish genes, our work predict important tuning of the expression of AGO protein and CAP1 protein to enable a robust genome defense. Because copy number expansion is a recurrent feature of diverse selfish genes we see across the tree of life, we envision that in many organisms, we see ampliconic selfish genes accompanied by co-amplifying suppressors. So within this higher level tuning of the general target non-specific defense machinery might be a widespread phenomenon across taxa, an essential strategy in the evolutionary battles against expanding selfish genes. With that, I want to thank many people, including my advisor, Alexei, who has been extremely supportive. I also want to thank my absolutely wonderful lab mates and 2 fantastic undergrad students that contribute significantly. Also, our collaborator, Grace Lee at UC Irvine. We just posted this work 2 weeks ago on bioRxiv, so please feel free to scan the QR code and check out our preprint. I want to thank you for your attention, and I'll be happy to take any questions you might have.
All right, so you've still got a minute or 2 for questions. So who would like to go? I know we had people sign in directly for this talk, so I know there are people here for it. All right, so Francisco, you have a question? Or possibly Blanca there, I'm not sure which of you.
Sí, soy Blanca. Muy buen trabajo, felicidades, muy bonito. Y bueno, pues los elementos transponibles, obviamente el genoma los está reprimiendo, como bien lo mencionaste aquí. Sin embargo, ¿crees que haya un momento de la evolución en que pueden ser motores de esta misma evolución? ¿Qué piensas al respecto? Gracias.
So I'm relying on the caption, which I'm not sure if it's
It was
translated
reasonably
correctly.
accurate. It's reasonably accurate.
But yeah, but I understand correctly, the question was whether transposon, the moment in evolution where transposon could be the engine instead of being the bad guys being repressed. I think it's certainly possible. It's interesting to know that in eukaryotes, there's no active mechanism to get rid of them, unlike in bacteria where we have CRISPR-Cas9 to cut out bad stuff. In eukaryotes, we only have mechanism to silence selfish elements. We don't have mechanism to get rid of them. So I think once we are stuck with them in the genome, we might as well. It's conceivable that we could find ways to make use of them. And the very fact that half of our own human DNA have obvious transposon origin, and many have been demonstrated to become sources of new enhancers or promoters, I think is lending direct support to that. They could be useful over a long period of time. That said, I think within a very short period of time, their acute mission is to proliferate, and that poses a threat to genome integrity. So it's really intricate play.
All right, and then we have a question from Spencer.
Yeah, so my question is about the stellate locus itself. So it's ampliconic and it's expanding. Is there some sort of equilibrium that you reach, or do you think this is a runaway process? And I guess the other question is, like, if you just are getting deletions all the time, so stellate's on the X, if you're just getting deletions, why doesn't that just kind of take away the process of the expansion of this locus?
Yeah, so I think there's some older literature that shows there's some variation in stellate copy numbers, and we don't really have more recent work, but I did a quick and dirty survey in certain lab strengths, and we actually find a somewhat bimodal distribution of the stellate copy numbers, which we don't fully understand why that is the case. So essentially, we have a bunch of strands that have low copy numbers and a bunch of strands that have high copy numbers. And actually very recently from J.J. Emerson's lab at UC Irvine, they put a preprint out from, I think, a few weeks ago. They just did a very careful analysis of stellate locus. And they also, again, they only surveyed, I think, 3 strains, but they were able to find one that has a very high copy number and one or two that have very low. So we don't know if this is truly a bimodal distribution in the wild, and we don't know what's the functional significance, what is the mechanism generating such a distribution, if it is real, but it's certainly very interesting and deserves more attention. And going to your second part of your question, why don't they just get deleted? Because I guess when you're repetitive or ampliconic, you have a chance to either expand on or delete is somewhat unstable. The short answer is I don't know, but I didn't really go into this, but there's there's a body of work that suggests that stellate is an X-linked meiotic driver. So they have this tendency to kill Y-bearing sperm to bias their own inheritance, the X chromosome that harbors stellate locus. So if that is true, then they have the ability to bias the inheritance of X chromosome harboring higher copy numbers of stellate. Because if you're thinking about silencing by piRNA, it's essentially a numbers game. So the higher copy number have a inheritance advantage, if that makes sense.
Yeah, absolutely.
All right, so I would like